Eric, you know what I like doing?
Skateboarding.
Nothing. But you know, when I'm trying
Second
to
best.
second to that would be fighting the existential dread I feel every day, and that comes in the form of lifting. And what is a more noble pursuit in lifting than getting absolutely maximally jacked? Is there any better pursuit? Self-betterment?
I would say the only better pursuit than getting maximally jacked is getting maximally jacked while trying to get minimum function out of it, trying to ensure that it doesn't benefit you in any other way, and that form does not follow function.
Yes.
That you are purely here to objectify yourself, and that's what we're about here at Iron Culture.
Yeah, we look. Everyone knows what a do-by daddy is. We don't have to go into details, but we want to be so massive, to be mass monsters, that it is offensive to other people, and that it causes problems for you in everyday life, such as walking through doorways, communicating without running out of breath, just those things. And for those individuals, wow, what a getting into the science of the hypertrophies, into individuals such as Brad Schoenfeld. Okay, Brennan Roberts, Juha. That's all I'm gonna say.
Juha Hulmi. Yeah, absolutely. I mean, if your goal is sleep apnea and an inability to clean yourself after using the toilet,
bowel movement.
then you're in for a treat, because we've got a really cool collection of three academics who are at, I would say, the forefront of thought in terms of hypertrophy science. Dr. Brad Schoenfeld is probably the most well-known, most prolific, who specifically did his PhD on mechanisms of hypertrophy and proposed probably the first robust model that is nuanced and includes some of the alternative explanations to just purely mechanical tension for hypertrophy, which over the last almost decade now, he has been a part of expanding and criticizing on his own. And two of those folks who've written papers with him, Brennan Roberts, done a lot of molecular work in hypertrophy and was on a paper specifically looking at issues with measuring actual changes in hypertrophy, which throws a wrench into all the gears. And then also out in the University of Jyväskylä, Dr. Juha Hulmi, who does a lot of very cool work as well and has done some work in animal models, looking at sarcopenia and hypertrophy in general. He was on the most recent kind of updated look at some of the signals and pathways to hypertrophy, and very much trying to bring nuance and potentially modify kind of the initial model that Schoenfeld has come up with. And we had a good talk about all the mechanisms. We really got into the weeds. So if you love science, this is definitely the episode for you.
Oh, yeah. No, I would say that this is an opportunity for individuals out there to listen to three of the leading scientists as it relates to hypertrophy, talk about hypertrophy, building muscle, what is the current state, what's likely next, what are the holes, what are the gaps that need to be filled. So if you want to know the science of hypertrophy, this is the episode right here, brought to you by Iron Culture Podcast. Wow, this is the Avengers when it comes to hypertrophy. If you want to build some muscles, I think we got four knowledgeable gentlemen that'll be able to talk on this topic. This is the antithesis, this episode, to what I affectionately call the shut up and lifters, who say like, bro, building muscle, the science of hypertrophy? Who gives a shit about that? Just go into the gym, do your 3 sets of 10, go to failure every single set, and you'll grow. This is the antithesis. Welcome.
Yeah, we going in.
We going all the way in.
Yeah. So first, I just want to say that I'm pretty excited because we've got a lot of doctors in the house, and that's pretty cool. And not only just doctors, but doctors of like muscles and jackness.
Yes.
So I'm going to go in kind of just some clockwise order here. We have Juha Hulmi from the University of Jyväskylä, which is the leading sports science university in Finland. I had the pleasure of meeting Juha in October last year, and doing some work, currently collaborating with a PhD student there, Ville Sola, who is doing some really novel investigations with fitness competitors and bodybuilders out there. So really cool stuff. A lot of cool stuff has come out of his lab over the years. Ahtianen's out there, of course. Komi, the late great Komi, just came out of there. and also Hakkinen. So a ton of great research coming out of this lab on concurrent training, strength adaptations, hypertrophy, and all kinds of muscle characteristics. So he's leading the charge now, and we're very pleased to have you here. Next, we have Dr. Brennan Roberts, who is works with the strength guys as a coach, but is also currently a postdoctoral researcher. He's supervising one of my students with me, and just a brilliant mind, very smart guy. Also competes in natural bodybuilding, powerlifting as well. Is that correct?
No, I don't lift heavy things, man.
Absolutely not. This is a hypertrophy podcast. That was a test,
size,
and you
bro.
passed.
It was a test, and you passed.
You passed the test. That's right. So so he's jacked, and he knows how to get jacked, and so it's been a pleasure. He's worked with 3DMJ. He's written a blog post for us and gotten on stage. So pleasure to have you here as well. You both Juha and Brandon have published with Dr. Schoenfeld, who is our of course the hypertrophy specialist, the
Godfather,
the magic man of muscles, the godfather of hypertrophy,
jacked Christopher Walkens.
The jacked Christopher Walken. Yeah, I mean, there's many titles, and he's earned them all. But yeah, we're privileged to have probably, I'd say that the current leading pioneer in hypertrophy research, especially from an applied perspective, which is cool because it directly relates to what we do in practice and all you Iron Culture listeners on. So first, Juha, tell us a little bit about yourself, your current research, and just introduce our audience to yourself, and we'll keep going around and get everyone familiarized.
Yeah, so thanks guys. Well, I think you said it all, but so I'm from Finland, and I was just recently appointed as associated professor. So
nice.
Yeah. So and I'm doing research with cancer models now. We're doing some cancer research and fitness research and resistance training research. Those kind of things are my main areas of research. So many things, not really good
awesome.
Focus. Yeah, and I also do some mentoring for some athletes, and I have a blog, and I'm doing those kind of popularizing science as well. So yeah, that's it.
Honored to have you here, man. Thank you. And Brandon, tell us what's going on in your world these days.
Yeah. So finishing up my postdoc for the past probably 3 years or so. I've been studying the molecular adaptations to resistance training. Background is in muscle wasting, though, so I decided, I'd rather gain muscle than lose it. So
good decision.
Yeah, studying that for the past few years. Found some interesting things. We're writing some stuff up now. Got to be part of a couple different reviews that were really fun. And then along the way, just applying everything that I'm reading to athletes, right? So bodybuilders and powerlifters, and also communicating a little bit of science through SciFit and, Skypes and blogs and stuff. So that's kind of it for now, and competing when I can.
That's right. Those who have joined us on video, yes, Brandon and I are starving, and yes, we are choosing to do that for the Instagram pictures primarily. So
I just want to note too that off camera, before we began, or off podcast, Eric, one of his first questions to Brandon was, "So are your glutes striated yet?" Was how he opened up his question for Brandon, just to give you some context.
Yeah, you ask the important things in life.
Yeah,
that's
not
right.
how you're doing. It's like, so glutes striated yet, bro?
That's the same question for a bodybuilder in prep. How are you doing? Is how striated your glutes. That's your gauge of happiness.
True.
So.
Fair.
And Brad, Dr. Schoenfeld, what is going on in your world? What's new?
Well, I'm the associate professor in exercise science at Lehman College, and I head their human performance lab. And what's new is we just got a master's degree program, and I am the graduate program director, so very excited about that. And as far as my world, I'm a educator, researcher, former bodybuilder, and personal trainer. So really the gist of my research comes out of who I was in a previous life. And I'm like a kid in a candy store that's just investigating all different things that I wanted to know while I was a personal trainer and bodybuilder.
Oh, I love it. This episode is going to be a discussion of C's candy. It's going to be just like Candyland for all of us. And Brad, I just want to say that I'm continually impressed by how much quality research you put out and how prolific you are, considering that only now do you have graduate students. Most people who have that kind of output have slave labor, and you're only just getting that now. So welcome to the real chapter of Schoenfeld publishing a lot.
And I will say that I am very fortunate to be surrounded with a lot of great colleagues, including yourself, who have collaborated on, and Juha and Brandon as well. So you surround yourself with good people, you can be very productive.
Love it. So I think, Omar, where I want to start, because I think if we kind of kick this off to really understand hypertrophy, we have to know, how do we even measure it?
Right.
Now, obviously, you and I measure it by looking in the mirror and flexing.
Bicep
probably
naturally.
Yeah. That's probably the most accurate way of doing it. But in the lab, where we have to deal with subpar methods of accuracy, what are the ways in which we actually measure hypertrophy, and how accurate are they, and what are their limitations? And I think I'll open this up to everybody. So whoever wants to kind of kick this off.
I thought Brandon, you want to take that?
Yeah. So I'll actually start opposite of where you would think we would start, and that's on the kind of muscle fiber level, which is probably my expertise. And the way we do that is we actually take biopsies from subjects or participants, whatever you want to say, and we can do that at different time points, before and after exercise. And then, much like a biology textbook, you could see that typical muscle structure thing, is the way we measure fiber size is literally just tracing how big the circle is inside your muscles. Now, biopsies they're safe and effective way to do it, but they're expensive and they're invasive, right? The other kind of problem that we've had is they may not be representative of the whole muscle. So if we take a biopsy, it's probably about as big as my finger, right? We shove it into the thigh or the deltoid or the traps or something, and we take a piece of that, and then we do some assays and stuff, and we measure how big the fibers are. Well, we're really only measuring, maybe the biggest biopsy I've ever counted and fiber typed and measured is probably like 2,000 single fibers, maybe 3,000, and where the average is more like 500 or 1,000 max. So taking that data and extrapolating it on the fiber level all the way out to like whole muscle size, right? You're taking a little piece and you're saying, okay, so we're going to extrapolate it out this far. So that's difficult in and of itself, because it's not really representative. We've done validation, and it actually does show some correlations, but I think the magnitude that you see the fiber level increase versus maybe a DEX or a ultrasound or something else is so much larger, and we really have trouble kind of explaining why that is. If you see a 40 to 50% increase in the muscle fiber size, you would kind of expect your muscles to grow to 40-50%, but they don't. So anyway, that's the kind of the baseline, super invasive, super kind of deep picture of muscle size.
Right. So we're talking an actual like a needle that is hollow that goes into a muscle and extracts tissue, and then you're looking at it under a microscope and actually looking at pre-post changes in cross-sectional area of individual fibers.
Yeah, for sure.
And interestingly enough, that doesn't agree with what seems to be happening when we look at things like whole body or whole muscle changes, or at least lean body mass changes. And do we have any idea why there is a disagreement? Like, what's the disconnect?
Oh, man.
And we're allowed to speculate here. No one's peer reviewing us.
Okay. I was like, I don't actually think we know. So there's, if we look at a muscle, there's a lot of stuff in a muscle, right? Mm-hmm. You have myosin and actin, proteins that actually contract. You have the mitochondria and all this other glycogen. Glycogen's a big one, right? Water retention, fluid. Muscle's actually, what, like 70% water? So I think there's so many parts that go into it. And, you know, the way we measure fibers is so particular. It's not like an ultrasound when you're measuring, you know, kind of a bigger image. So I think just the way we measure it is just kind of inherently, like, we can detect larger changes. And that's what I wanted.
Got it. So Juha, Brad, any thoughts on some of the disconnects in different measurements?
I think the pennation angle is one thing. So we are measuring with fiber size. It's this physiological area, and then anatomical area is a different one. So the anatomical one is the one that we really see. So when you flex your muscle, you can see that in the mirror. But then fiber size, we usually try to align the sample in a way that it's cross-sectional. So then it may be a bit different than the one that we can see at the whole muscle level. So that may be one thing. Maybe that can explain some percentage of the thing. So because when we train, this angle of muscle fiber is changing, and that may explain some part. I don't know, but that's. I have read the review of you guys, and I think it was a really good one, and I know also that we have seen the same results, that the fiber size change is quite huge, and then the anatomical change, you know, like MRI, ultrasound, and so on, those kind of things, they are not changing as much. So I can only speculate. Maybe some fibers are even lost. I don't know. Interesting.
So for the listener, can you explain what a pennate muscle is and what pennation angle is, so they have a better idea of how that could play a role in the actual changes in total muscle size versus fibers?
I don't know if I'm best to explain that. We measure pennation angle here.
Just the very basics, because I think some of our listeners might not know what pennation is or pennation angle is.
So maybe half of the muscles don't have this pennation angle, so the fibers are just starting from the tendon, let's say like biceps brachii. So those they don't have any. Those fibers don't have any pennation angle, so they are straight. The fibers are straight from the tendon to another tendon. So quite easy to imagine that kind of a thing. I think most of the people think that that's how muscle fibers look like. But on the other hand, quite many muscles have this pennation, so it means that, for instance, vastus lateralis at the quadriceps, one of the most famous muscles in quadriceps femoris, vastus lateralis that we usually measure, it has pennated muscle fibers, meaning that the fibers, they don't run from the tendon to tendon, but instead they are aligned like pennated. Maybe you guys can explain it better.
We're talking about Kai Green's quads, bro. Those cross striations when you see him on the side coming in when he's shredded. You can see that the lines of the fiber connect kind of laterally across down at an angle. So the pennation angle is actually the change in that orientation with weight training that happens, which gives you a slight mechanical advantage, and it changes how much myofibril packing density you can get, how many of those myofibrils you can fit into that muscle area, which means the cross-sectional area is going to be influenced not just by what's the total amount of mass, but the angle that it's fit in there, and then how you measure it. So, which is a kind of a cool thing that I remember learning as an undergrad, and then again as a postgrad. It's like, what? That changes too? Like the angle that my fiber attaches into the tendon, which I just think is really neat. And then I started thinking about Kai Green's quads, and then I looked up and I'm like, oh shit, I missed 20 minutes of lecture. So I don't know if you guys had that problem.
I'll give you an aside there. One of the reasons that it's postulated there is a disconnect between strength and hypertrophy is because of the increase in the pennation angle. So the less pennated, the less of an angle there is, the more in line with the fiber is with the line of pull. So even though you're able to pack more fibers in, you're now not having the same line of pull, and it's thought that reduces the ability to commit force, to have strength in a muscle. So ultimately, there's at least some theory that that's why you don't see this real linear relationship between increasing muscle size and increasing strength. So you do get more ability for sarcomeres to shorten, but you're having a reduced angle where it's optimal for the line of pull.
Right. So you're kind of trading mass for efficiency to some degree there, right?
Exactly.
It's fascinating.
Very interesting. Very cool. So, and we've got other methods to measure this stuff. What is it like in an ideal world? Let's say you've got a $20 million lab and you want to investigate hypertrophy. What would you guys have in there? Brad, I'll start with you. What do you think are some of the best methods?
Yeah, MRI clearly is the gold standard measurement, but you have to also remember there's differences. The ultrasound has very high correlation to MRI at a cross-sectional area. So MRI, ultrasound, even with muscle thickness, which is a single site measurement of a muscle, if you look at that, it has a very high correlation to the cross-sectional area at that specific site. What it does not have a great correlation to is the volume of the muscle, which really is looking at the total growth along the full belly of the muscle. And here's the issue and this has mostly been seen in the quadriceps muscles, which have been shown to have non-uniform development. And a lot of times, different muscle actions, so eccentric versus concentric actions. So with an eccentric action, you're going to tend to get more distal hypertrophy, whereas it's more midpoint to slightly proximal in your concentric actions. And even just different, an exercise program itself will result in non-uniform growth along the muscle. So if you're going to measure with MRI, where you're doing serial slices down the length of the muscle, and look at the volume, so the totality of what the muscle is growing, you're going to get better results than you would with, let's say, an ultrasound or even a CT that's either a CSA cross-sectional area or a thickness. Now, I will say there's ultrasound now we now have 3D ultrasound, which is a newer technology that has the potential to be really amazing for the measurement of hypertrophy. It's still somewhat of a new technology and is more evolving. But I would say that really any site-specific measurement, and I think we can, other than the biopsy, the histological analysis that Brandon brought up, we can kind of categorize the other methods into non-direct methods, indirect methods, which would be DEXA, underwater weighing, bioelectrical impedance analysis, even skin fold analysis, BOD-POD. Those would be your indirect measures versus a site-specific measurement, which would tend to be more, and it's research actually shown this, more able to pick up subtle changes in muscle growth, such as the three primary ones would be CT scan, MRI, and ultrasound. When you then look at the indirect measures, such as DEXA and underwater weighing, there's differences between those as well. Like DEXA, you can there's ways to estimate actual muscle changes, where underwater weighing, you're basically looking at just fat-free mass or lean body mass versus fat mass. And you do get, with both bioelectrical impedance analysis and with DEXA, you get some additional ability to look at least regional changes in certainly lean mass and to some extent muscle mass. So they are estimates, they're less accurate. And the accuracy that we've actually done some validation studies, the accuracy on a group level, when we're looking at a group of subjects, let's say we're looking at 30 subjects, the accuracy of DEXA and bioelectrical impedance and BOD-POD, they actually have very similar abilities to look at lean mass changes, overall lean mass changes. When you start getting into the more regional specific, that's where your ability becomes less. And certainly if you're just some dude who's jacked, like, you, Eric, and you say, I want to get my DEXA and see, there's going to be big differences then between BIA or very, I say big, substantial, where it really is not providing more than gross indications of your development.
Right. And I think that's really important to understand the difference between these direct and indirect measurements. And the indirect measurements are very much like body comp assessments versus muscular assessments often, and there was a cool paper that came out not too long ago showing that for every change in a unit of fat mass, there's an obligatory change in lean mass, because some portion of adipose tissue is actually lean tissue, around 13% if you read Hall's paper. So that means if you're gaining a ton of body fat or losing a ton of body fat, you're always going to show quote unquote lean body mass losses, when you look at something like a DEXA. However, you might do an ultrasound scan and see no change. And I think that's something I didn't realize when I was first reading papers and trying to disentangle, what's going to cause muscle loss, what's going to cause muscle growth, and how do we apply this, and like the reviews I first wrote when I was getting involved with the peer-reviewed literature. And then actually conducting research now and seeing the disconnects, it makes it even easier to see why we can have some head-scratching outcomes in papers where there's a disconnect between strength and hypertrophy, or we see, a loss of lean body mass in a dieter but no loss of strength. And I think that's something that the casual science consumer may not understand. So I really appreciate that perspective, guys. So anything else you guys want to add on the measurement side of things before we start talking about what the heck is going on when muscles grow?
One thing I would like to add is that we cannot measure muscle mass in humans because we have to investigate live humans. Maybe in the Germany in the 40s you could investigate muscle mass from humans, but that's not possible. So we can measure muscle mass from animals if we do animal studies. So that's what we can do. Yeah. And
that's actually a really good point. So what have we learned from animal studies that's informed what we know about the way muscles grow in humans, and then what are some of the, how far can we trust animal studies before we start extrapolating too far to people?
So I'll start off with that one because I have a little itch. But so I've done animal and human research, and I think going back to Lexel's traditional paper, I think it was in the 90s or whatever, early 90s, late 80s, when he looked at just counting the number of fibers in vastus lateralis, so a thigh, right? Mm-hmm. And he quantified it in like 15 or so people. And he said, okay, so this takes a lot of work, right? And that's where maybe they started to think, well, we can't quantify muscle mass in live humans. We can do indirect measures, or we can take pieces. But maybe we don't know if muscles are getting bigger, or maybe they're just like almost multiplying, right? So that goes to the hypertrophy versus hyperplasia. Which,
can I add one thing?
Yeah.
So they were dead. So they were cadavers, the subjects.
Yeah.
So no ethics needed on that one, right? You don't need to get informed consent.
Yeah. They were already dead. Yeah.
Sorry. Say nothing if you agree to be in this study. All right, John Doe, you're in.
Yeah. Sorry for the interruption. Yeah.
No worries. That's a good point. And it was mostly in males. So if we look at the aging kind of fiber loss literature, it's kind of like you're like, something else is going on there. But anyway, so when we look at animals, we can stress them. We can take out a muscle to cause other muscles to hypertrophy, which is synergistic ablation. So like if you took out a piece of your calf, right, the other part would get bigger. And that's not really translatable to humans because we're not taking muscle out. That's, it's hard to rationalize that one. But it can tell us lots of mechanistic things. And I think that's where the animal literature really shines, is I can't take things out of you, and it's really hard for me to put things in you that aren't approved drugs. So that's how we're going to tease apart and find out that mTOR is important or AKT is important. Pick your molecular marker. But there are some differences, and I think going back to the hyperplasia thing, there's a review and then a meta-analysis showing that hyperplasia can definitely occur in animals, especially like birds. But there's only one study in humans, and so we're not sure. So that's kind of those two type of things, that how you can use animals, and then maybe they don't relate to humans, but maybe in extreme cases with bodybuilders with lots of drugs, maybe they do, and you can hyperplage, but we don't really know for sure.
So Brandon, just for our audience, what is the difference between hyperplasia and hypertrophy of fibers?
Yeah. So hypertrophy would be kind of I talked about earlier. So that would be one cell. So a muscle fiber is a cell, right? And that would be it growing kind of in size, right? So you're getting thicker, your biceps getting kind of thicker. Hyperplasia is if it split into two pieces. So you've got one cell splitting into two, and then it started growing. So again, I'm not going to say it doesn't exist because I kind of lean that it might in extreme circumstances, but in animals it doesn't translate as well to humans.
Got it. So it seems like, and that could just be a measurement thing because we can look deeper at animals versus and we have a larger body of evidence looking at actual biopsies and dissection of animal muscles versus something I was talking to Andy Galpin, and he was just kind of expressing the very few number of studies we have on high level athletes and resistance trained individuals where we've actually done biopsies, and especially in the last decade of them, and looking at the actual fiber types. So I think that's something we probably don't really know, or don't realize how shaky that evidence is. I think we love talking about fiber types in fitness and in bodybuilding, but we basically have like a handful of papers that are almost always super underpowered that are actually looking at like resistance trained people. Like, people go, the fiber types of bodybuilders, powerlifters, and weightlifters, like that's like one study. I think there's maybe two studies where they've actually looked at that, and they had like N of 5. I think that's a really interesting point and good thing to point out.
Quick aside, I could be totally off base here to ask Brad. I thought I remember from his Can't Fit Pro talk several years ago about hyperplasia. Is there any theoretical role that PEDs can play in that process? What were you saying about that, Brad?
Yeah, so the theory is, at least one of the working theories, is that when a muscle reaches a certain critical threshold, it will then basically, it has no choice if it's going to grow but to split off into two. And that would be similarly like Brendan had mentioned, the synergist ablation model. You see massive hypertrophy. You'd see 40, 50% growth in a matter of weeks in a lot of cases, where the theory is that and Joey Antonio's old work in quails, where he taped up their wings and added little dumbbells to them and had them walk around for 30 days, like this in a loaded stretch. And so again, massive 300% growth. That at a certain critical level, the fibers will have to split to continue to grow. And the theory would be that PEDs might be able, if you do enough of them at least, to get past that critical threshold where it would cause that. Now, that is not by any study I've ever seen. You're certainly not going to get an IRB at this point. Maybe I don't know what country you might need to go to get that done. To really study something like that. And if you're looking at it in a bodybuilder or bodybuilders that are already doing them, there's all sorts of confounding factors. So you're not going to really know whether they actually, because you're not going to get them before they do it and then after. So really it's a working theory, but that would at least hypothetically be a rationale whereby in humans you'd be able to do it. Or if you're just a genetic freak like Eric Helms, where you just massively grow, that could be a case too.
I'm predicting that's the second compliment he's given me that Brad is going to ask to borrow money off camera after this podcast.
No,
is over.
There's a pyramid scheme after this, the muscle pyramid scheme. Interestingly enough, that
tell you what,
it's total respect, Eric. That's all.
I'm one compliment away from you being able to get whatever you need out of me. So
he's opening up.
Jam show.
Some people need liquor. Eric just needs validation from others. So it's just
100%. I mean, like I don't really have any like self-worth internally, so I just need it from others. So I really appreciate this. You're literally keeping me alive.
Hey, before we go on, just quickly, a few more definitions. Brandon, you said mTOR. Can you guys just talk about what that is?
So I'll start out. So mechanistic target of rapamycin. So most of your listeners are probably somewhat familiar with this, but it was shown back in the late 90s, I think. That it's
92 by David Sabatini.
Sabatini
in animals, yeah.
Yeah.
Yep, Juha won that round. Got the citation.
I was going
Boom.
with
If this was Jeopardy, yep, we got a winner here.
I was going with the Bar
Sorry.
and Esther 99 reference, but anyway, yeah. So it was shown through a series of studies that mTOR is a player in muscle hypertrophy, such that if you use rapamycin and you kind of stop it from working, simplistically, it will stop the hypertrophy mostly from happening. So that we do a series of gain of loss and gain of function, I think, loss and function, to show how things work in the molecular adaptation realm. So that was just a few experiments back then, and maybe Juha can elaborate a little bit more to show that mTOR is important for hypertrophy.
Yeah, that's pretty much it. And some recent studies have shown that it's also important in relation to resistance training adaptations, both in animals and also recently in humans. So it's possible to give this drug rapamycin also to humans. So Blake Rasmussen's group has shown that in humans. So it blocks resistance exercise-induced protein synthesis if you give this drug that prevents mTOR activation. So it also works in humans. So it seems to be important, one of the most important regulators or hubs, signaling hubs for the adaptation. So it sort of transmits the signal to protein synthesis.
I'd add it's generally considered a molecular nodal point. It's kind of a midstream, so it's going to take signals from your upstream regulators, and there's multiple pathways that feed into mTOR. There's actually two different mTORs. It's really interesting. We talk about some of these things, but we're still, I would say, in somewhat the infancy of really understanding. As much as we'd like to think science has taken us so far, and it has compared to where we were, we're still so far from knowing all the nuances of this. But mTOR is this molecular nodal point, and there's mTOR C1, which is the primary regulator that's been well studied. But interestingly, within the past few years, it's been shown that mTOR C2, which is the non-rapamycin version enzyme aspect of mTOR, actually has an important role as well. So if you knock out mTOR C2, it also will blunt the hypertrophic response. And there's other mTOR independent pathways that potentially can get you downstream to protein translation. So it really, as I think my colleagues were intimating, it's very complex. The pathways, if you look at them, if you were to map pathways, I do talks and I show like a simplified map of pathways, and it looks like 50 LA expressways kind of converging into each other and bifurcating. So it really is, I think, a future area that we're going to really start to understand the full aspects of how muscles grow.
That's really helpful, guys. And I think just for the listener, just so you kind of understand what some of this molecular work means and the way it operates, is essentially everything. Our bodies are these machines that take some kind of physical or mechanical stimuli and then translate into something biochemical. So when we're talking about lifting weights, it's actually quote unquote mechanotransduction. It's generating tension within the fiber as we move, and then if there is sufficient stimuli, that is translated biochemically in through these molecular pathways where things are actually happening. There's keys attaching and unlocking enzymes and proteases and all kinds of stuff that initiate adaptation. And the way what they're talking about when they're investigating knocking out, quote unquote, one of these things or targeting it is you can give a drug in an animal or sometimes a human, again, depending on IRB and what you're allowed to do and what it'll do to them long term. It will block one of these pathways, and then you can see how that affects the downstream adaptation. And then okay, everything else is the same, but I've modified this one step, and therefore we know what this could be a rate-limiting step for, or allows, or blocks, or whether or not there's some other secondary, tertiary, or quaternary thing that still allows it to happen. And that is kind of the craziness of the human body, is that it's been layered over through adaptation over millennia. That you can block one pathway and things still seem to work. And man, we're learning new pathways all the time. The pathways that we're aware of now in 2019 is a multitude more than we knew in the 80s. And we often make the mistake in science in thinking that we've got it all figured out, and then we block the thing that we think is key, and it still works. And we're going, well, shit, that means there's got to be something else going on we're not aware of. So that's kind of this it's almost this whack-a-mole of pathways that keeps popping up in this field. So it's a very, like Dr. Schoenfeld said, it's an emerging field. We're just learning this stuff. And through the combination of looking at the mechanisms and then the output, we're able to slowly get this kind of emerging murky picture. So I'm going to use that as a nice dovetail, unless anyone else wants to comment on that. Yes, Dr. Isuf.
What I was going to say is the Broseph himself. I think, because I know Eric's questions that he has lined up, which are super juicy, and it's going to be at the bleeding edge of what's being discovered when it relates to hypertrophy. So all listeners are on the same page. And I know it's changed over time. Can we just briefly discuss the different mechanisms of hypertrophy and how we've understood hypertrophy or those mechanisms to change throughout time, where what we understood in the 80s, 90s, 2000s is now currently? So just an overview maybe for like 10 minutes before we get into those questions about
absolutely
muscle damage.
Let's take a trip back to when Omar and I were little babies, and maybe I'm not going to state any names, but some people might have been adults in this conversation. What do we think was going on?
We know this. That's why you see that.
Correct.
You think
he is an immortal vampire.
Yeah. You're obviously referring to Brandon right now.
Yeah.
Correct.
Yeah,
Correct.
I'm actually 78. Yeah.
Yeah.
I thought it was 7,080, so that's pretty good.
Dude, that Finnish fish
78,000
keeps you
years.
alive. Correct. Yeah. So we have a vampire. We have Brandon, who just doesn't age from constant contest prep. Yep. He's actually 60. And then Dr. Schoenfeld, I think, is around 23? 23?
24.
24.
Just turned. Yeah.
The Doogie Howser of the group.
Doogie Howser professor.
So where were we in the 70s and 80s with our understanding? And you guys may not know this, but basically, what is your awareness of the first kind of model of why hypertrophy happens? Where did we start in this realm of research?
Well, I'll kind of start off that there was studies by Goldberg's lab, very famous studies, and they basically were the ones that came up with the theory, which is now a very well-accepted theory, that mechanical tension was the primary driving stimulus of hypertrophy. And they actually, interestingly, had speculated that the hormonal effects were minor. And then after that, towards the late 80s, early 90s, came the whole hormone hypothesis, stating that the post-exercise increase in anabolic hormones largely regulated the accretion of muscle mass. And that was the whole push towards doing higher, basically bodybuilding style training. So moderate repetition, short rest intervals, multi-joint exercises, multiple sets. And then, as hopefully we all know, that now has, largely due to Stu Phillips' lab at McMaster, there's the body of research. I'm not going to say it completely debunks it, but certainly it would suggest that any effects are minor if they do even exist. There's really not much to that theory.
So yeah, that's a really interesting one, where this is a case in science where we kind of had it figured out pretty early on, and then we followed a red herring of these. I think it's also important to put in context these small increases in hormones. It's not to say that growth hormone or testosterone taken as like exogenous performance-enhancing drugs in huge amounts by bodybuilders won't affect you, but it's that mechanical tension after maturation, once you're no longer going through your growth spurt, is the primary mechanism of growth. But of course, if you're an IFBB pro, inducing a second growth spurt while you're lifting weights, maybe the lifting weights is not as the only element that's going into growth.
Super physiological doses of the good stuff. Well
said, yeah.
And by the way, to that point, Eric, even within chronic, let's say, testosterone levels, I have not seen compelling evidence that if you're within the physiological range. Now, maybe if you're at let's say, so the physiological range usually is around 300 to 800 or 1,000 nanograms per deciliter. if you're at 300 versus 1,000, there might be some difference. But if you're 400 versus 600, and that's a 33% increase if you go up, I'm not convinced that there's any relevance to building more muscle at 600 nanograms per deciliter constant versus 400. And testosterone is very labile anyway, so it's not like you're always at a given level.
Yeah, I think even it might have been Krieger who did this analysis in the weightology review. He looked at all of the HRT and TRT stuff within the normal range, and I think he concluded it was like half a kilogram of lean body mass for every 100 step up in testosterone in the normal range. So exactly, we're talking about something you wouldn't even notice the difference of. It would be like point something on the FFMI for going up from low normal to high normal. It's a very, very modest change at baseline. And it is interesting that it is the baseline too. Like these changes in your standard level of testosterone is going to be like where you're at before you even lift. And it may not even predict what happens when you start lifting, which I think is also quite interesting. So yeah, it's not that testosterone doesn't play a role. It's that these small like diurnal variations level of kind of changes, the kind of changes you'd have in growth hormone just from being awake in the morning is similar to what you might see post-workout from doing, squats and deadlifts and stuff. It's unlikely to kind of have the same effect as mechanical tension. So it's about putting it in perspective. Would you guys say that's pretty accurate?
Yeah.
Yeah. Okay, cool. So we got a little away from this model of mechanical tension being kind of the underlying stimulus, if you would. We got mired in what I would call a red herring, and kind of focusing on the post-workout anabolic milieu from training. And how do we get steered back? What is some of the research that you mentioned was done out of Stu Phillips' lab at McMaster that kind of brought us home and made us realize, oh, actually it was and still is the mechanical tension?
Well, Stu's lab really didn't show about that it was the mechanical tension per se. They just were able, they did a model, they used a couple different models, but the basic one was doing a within subject design where one leg was subjected to one stimulus, another leg to a different stimulus. One leg was subjected to a high hormone stimulus, the other to a low hormone stimulus. And basically it showed no difference in the growth. So whether it doesn't necessarily show that it's all mechanical tension, but it does show that the hormones were not, at least that's the causal, you can try to draw causality where those would lead you. Now, I will say too, in fairness, there was a study by Ronestat, and I know that's been somewhat controversial, but that did show some differences. He used a somewhat different model than Stu did. We used the same model, but he did the high hormone
reverse
after
order.
the low. Yeah, right, exactly. So, these are things that need to be teased out, I think, to really get a pure determination of it. But I think in both of them, even if you look at the totality of evidence, and Stu's done a lot of large correlational studies where he looked at basically correlational data cannot be used for the most part to draw causality. And that's, by the way, that was the real issue, that the whole hormone hypothesis was based purely on correlational data. It was never based on any hard data. And as we know, you can draw correlations between eating ice cream and the murder rate. So ultimately, I think the evidence out of Stu's lab, in my humble opinion, really, I don't want to say debunk, but it really throws a lot of holes into that theory. And in my humble opinion, makes it where I would not use that as any, hold that up as any real way that you can gain muscle, substantial muscle. And Juha, Brennan, you.
Yeah, I'd love to hear it. Let's either one of you who want to chime in, anything else you have to say on kind of this evolution of our understanding of hypertrophy?
Yeah, I think so. That's all spot on for sure. I think tension is kind of well known as the primary driver. Since that period, I think something that's come a long way, and I'll switch a little bit, is the metabolic stress aspect, just because of the BFR training, and that is definitely secondary to tension for sure. But I think even in the past few years, we've started to figure out just how much metabolic stress, where we thought it was important, maybe it's not as important. It's kind of another one of those hormonal type things where we're just still figuring it out.
Can you guys just quickly define, mechanical tension and then metabolic stress?
Sure. So mechanical tension is like stress on a muscle. So like you lift the weight, it causes stress on the muscle, tension, contraction, right? So that would be mechanical tension. Generally elicited by higher loads, just when you think of it in general. So metabolic stress would be kind of lactate, reactive oxygen species, other hydrogen ions that occur through more like pump work, as we'll call it. So those are the main two differences.
I think it's important to point out that we're talking about tension that's experienced at the fiber level from work. You moving. Every time you contract the muscle, there's some level of tension, but there's probably some kind of threshold or level of sustained tension at a high enough amount that produces an adaptation in muscle. And then metabolic stress is we're talking about the byproducts of taking that mechanotransduction and making into a biochemical signal and producing muscular work, and that potentially creating a metabolic environment in the muscle which produces stress. So they're intrinsically linked. Would you guys say that's accurate?
Oh yeah.
Yeah.
Which go ahead, Brandon. I know we got bogged down in the definition a little bit, but important. No, keep going.
I think that was it. I'll just introduce the last one, and then we can open it up. So, Brad's seminal paper, I think it's got over almost 800 citations now, is tension, metabolic stress, and then muscle damage are the big three, right? And they've evolved over time. I mean, again, pretty clear that tension's the number one. Metabolic stress is important for sure. We don't know exactly how much. And then damage is actually, I was reading, kind of brushing up a little bit, has been shown to be a little maybe less important recently. So I don't know if you guys want to pick any part of that and kind of expand, but.
So
you got any thoughts on this?
Yeah, we have this history with Brad. We invited Brad to talk in Jyväskylä 2017, and we met also. So I organized this symposium. We had one day of mechanisms of hypertrophy, and so I
just invited. 1 day?
Yeah, just 1 day. But anyway, so we had some talks about how this muscle hypertrophy is censored and also stimulated biologically. And then this Henning Wackerhage, professor in Germany, he emailed us after the meeting that maybe we should write a paper about this topic. So the stimuli and sensors that are regulating muscle hypertrophy after resistance exercise. So we said yes. So we were like 5 of us, and we started. Okay, this is not that difficult. There are 5 of us, and we just start to write. I think it took like 50 versions of the review, and it took like almost, I think, about a year. We just wrote it, and we tried to find evidence for all of these things, like mechanical stress and metabolic stress and muscle damage, and just continued, and we couldn't really stop it. But then finally we submitted it, and we got it accepted a few months ago. Now it's published. So those people who are interested about those molecular things, you can look for this paper that me and Brad and others wrote. So it's in JAP journal this year. So
and if I can get up on my soapbox a little here, I think this is really important. The longer I've become a researcher, and I came from a personal training background, and I was very quick to jump on bandwagons and be opinionated. In my humble opinion, that's all I can speak on, the true researcher and scientist is very cautious in terms of drawing opinions. And the more I submerge myself in the literature and in carrying out research, the more cautious I am to draw strong inferences without very strong supporting data behind it. And I will say this, I've seen many people in this field come have very strong opinions. And look, it's going to get you publicity. You can write blog posts and get by saying, it's all mechanical tension. The data, and look, no one knows more than Henning and myself and the rest of us who were on this paper. And so we submerged ourselves in this literature. There's no one that can draw strong inferences based on the data that we currently have. I can argue both. If I want to be a lawyer, I can argue both sides of the case and make it. And you'd come away saying, if you weren't totally submerged in the literature, you'd come away either saying metabolic stress is completely necessary, or metabolic stress has no bearing whatsoever on results. I can tell you there are a lot of. And also, to take a step back, one of the real issues is you can't study this, at least there's no way I can figure out, and no one else I know has, without having a huge amount of confounding variables. So to try to draw causality on these topics becomes very difficult. So if you want to, let's say, look at just muscle damage, well, yeah, you can do eccentric training versus concentric training and have more muscle damage, but then you're also dealing with the different aspects of eccentric actions versus concentric. You can use heavier loads versus lighter. Anyway, there's all sorts of things you can do, but then how do you tease out those factors? I can tell you that from a muscle damage standpoint, as Brandon was mentioned, I want to point this out because we wrote a letter to the editor. It's on my blog. Those people can read it, with the Damas group, who published a review paper based on their previous study. I'm sure most people listening or many have seen it, but if not, there was a paper by Damas, headed by Damas Felipe, and it was a very good paper. I'm not trashing the paper in any way. And the paper was designed to show, they were looking at the time course between protein synthesis in the initial stages versus at the latter stages. So we had known that if you look at the initial stage of when you do, let's say, a biopsy and look at muscle protein synthesis in an untrained subject, it does not correlate with the hypertrophy that you see over the long term. And what this paper did, and it did it very eloquently, was to show that the reason, or at least the perceived reason, was that in the very initial stages, you're going to see larger amounts of muscle damage in an untrained subject. And then they get this repeated bout effect. You keep doing it over and over. The muscle damage attenuates. So in the initial stage, you see very high levels of protein synthesis. And their theory is that this is all then, or much of it is directed towards repairing the tissue instead of creating more muscle. And they drew this is the issue that I have, is that they then took it and said, well, this is evidence that there's no effect of muscle damage on hypertrophy. Well, there's no way you can draw that. If you actually read the study, it was not designed to show that, because the muscle damage theory is not you do one session and get a lot of muscle damage, and that's going to drive muscle growth after 10 weeks. It's that if you get serial levels of moderate muscle damage, this is at least the working theory that we espouse, and whether it's true or not, we don't know. But at least hypothetically, you can make a case that's moderate amounts of muscle damage over prolonged periods of time might help to drive the hypertrophic response. We know that it's involved in the COX pathway, which has been shown to be a somewhat critical regulator of satellite cell activation. And really, that might be the primary way that muscle damage over time elicits greater hypertrophy, is through initiating greater satellite cell activation and perhaps proliferation and differentiation. It also increases the activation of macrophages and other, which have been shown to potentially have hypertrophic effects. So can I say that muscle damage has an effect on muscle growth? No, it's again purely speculative. And I would say that there's other evidence that it does not have an effect. But we certainly cannot take that leap and make strong conclusions either way on that or on metabolic stress. The same thing. I've heard others completely dismissing it. And there's just recent study came out a few months ago showing that if you infuse, or that the scientists infused lactate into a rodent model, they had rodents exercise at a submax level, aerobic type exercise, so there was not much lactate buildup. They infused a physiological state, which is basically seen in resistance training, so an amount that would be conducive to what you'd see in a resistance trained subject. And they actually showed that the group, the mice group that got the infusion of lactate had much greater hypertrophy, significantly greater hypertrophy than those that didn't. Now, could there be other confounding factors? Of course. They've shown in test tubes that lactate is anabolic. Here's one last thing I'd like to leave you with on that. And I think this is a really interesting area of speculation, at least with metabolic stress. Fast twitch fibers have been shown to have a reduced capacity for producing force with the buildup of acidosis. So we know that metabolic stress is largely attributed to training in a glycolytic environment where your fast glycolysis is prevalent. You're getting a buildup of lactic acid. Fast twitch fibers have been shown to not be able to produce force, so it diminishes their force output. Well, it leads to speculation that, let's say in this higher rep range, training at a higher rep range, if you're deactivating, if you will, even though they might be recruited, the fast twitch fibers, they can't really be stimulated to a greater extent because they're not able to produce force due to this acidotic buildup. And thus the type 1 fibers would be working more and thus potentially have a greater potential for hypertrophy. This has actually been shown recently in a number of studies. There's been 2 that I'm aware of, and I think there's a third one coming out, but 2 out of a colleague of mine out of the University of Norway. I think you know him too, Juha, and the Norwegian Sports Science Institute, that showed much greater type 1 hypertrophy, both in powerlifters and in just regular non-training subjects, recreationally active subjects, of type 1 growth in blood flow restriction training. Now, is that due? So again, we can't draw causality on these. This is why I'm certainly not then saying that this is real proof of anything. I'm just saying that you look to triangulate evidence when you try to draw inferences, and triangulation is a strategy where you say, all right, we're going to take, let's say, acute data. So you might look at EMG data, that wouldn't be relevant here, but then you might look at the acute muscle protein synthetic response. Then you look at the cross-sectional area based on MRI. Anyway, you look at multiple aspects. So if you see that an infused, let's say lactate into a test tube is causing anabolism, if you're seeing that infused lactate into a rodent model is causing greater hypertrophy, if you then see that in vivo, in humans, that in, let's say, blood flow restriction, you're getting greater type 1 growth, I mean, you try to triangulate and say, hey, look, maybe there's something to this. So I know I talked a lot, but I think this is just very important for people, because most people aren't scientists. They're not entrenched in the literature, and they hear someone who has good credentials, and they're people I respect, many of them. So I'm not trashing them. I'm just saying that it's, to me, really important to take a step back and not be so, not be ready to jump on bandwagons so quickly.
Yeah. So when we wrote the review, we wanted to have like pros and cons for each of these arguments, like mechanical, metabolic, and damage. So for instance, for this damage, we wanted to find whether there are any direct evidence that damage per se would increase muscle size. And there are some evidence that with some drugs that induce muscle damage per se. So there is no exercise, no metabolic changes, just the damage. That can induce muscle hypertrophy in some of the fibers, for instance, suggesting that damage per se can induce hypertrophy in some cases. But if you have too high or too large damage, then you can also induce atrophy, so decreased muscle size. So it's really dose response as well. So we wanted to look at those kind of things, and then also, like Brad said, many of the pathways and those kind of things, whether they could really affect. So for instance, when there is this muscle damage, there is this stem cell activation, satellite cell activation. And there is some evidence that if we just inject satellite cells to muscles, we can induce them to grow bigger. And we know that this muscle damage inducing exercise is the most potent stimuli of satellite cells to be activated. So there are some situations maybe when muscle damage is potentiating muscle hypertrophy. Perhaps the mechanical stress and metabolic stress is not sort of optimal. So in that case, damage can still potentiate. But let's say we have a situation where we have optimal mechanic and metabolic stimuli that drives the maximal anabolic signaling. Then we can know that there is no chance to even more upregulate those pathways if we have more damage. I don't know if you know what I mean. Mm-hmm. So, for instance, there are studies where people have been investigating this blood flow restriction by just walking. So people have been just walking and then using these cuffs, and that can even induce muscle hypertrophy, suggesting that perhaps
at least in the untrained.
Yeah, so perhaps the mechanic tension alone cannot explain that. Of course, there are some fibers that will have this mechanic tension, but perhaps there is some metabolites that could be in those situations inducing hypertrophy, even damage. We know that there can be even some damage in some fibers. So just to know, just what I wanted to say here is that in that situation, definitely the mechanic loading is not optimal, but still we can get some hypertrophy because we are combining perhaps these three stimuli. But when we are doing high load training with very low metabolic stress, let's say eccentric exercise that doesn't induce lots of metabolite accumulation and so on. So in that situation, perhaps the mechanic signaling and damage signaling are so highly activated and sensed in muscle that we don't need so much of the metabolite accumulation anymore. So in that situation, it's a different one than when we train with the lower weights and higher rep sets. So then the upstream sensors and mechanisms are probably a bit different than with different types of exercise. So that would be quite interesting if we figure out eventually these sensors and signaling. Then we can perhaps understand also this training a bit better. So we can say that, okay, perhaps in this situation we should train this way because we know that that's the way we can maximize the signaling and maybe in injury situation we know that, okay, now you cannot train this way, so then you can maybe hit these kind of pathways with the lower weights and so on. So I think that's applied side of these biological studies as well.
Brandon, you got any insights here or things you want to
add? So I think I'm going to go a little bit more big picture. So if we think about the field as a whole, right, there are a lot of researchers who are very interested in specific aspects of it, right? You've got STU and muscle protein synthesis. You've got, DOMAS and muscle damage, just a couple examples. And what I just realized this as I was listening, I was like, I wish we would try to tackle one question at a time. But that'll never happen. So what we do, like Brad says, is we take pieces and we try to make a picture. But, science is ever evolving. So we're constantly figuring the picture and saying, actually, that piece doesn't go there. It goes over here. It's not even on the board. And another thing we do is we do a lot of research in untrained people because when you start doing research in trained people, they have varying backgrounds, right? So it's a lot harder. There's a lot of other factors too for recruiting trained people. So translating, the science of an untrained person, most people can do pretty much anything and gain like 3 to 5 pounds of muscle in 12 weeks or 16 weeks or 20 weeks. But then taking that next step, right? That's where probably most of your listeners are at. They're like high intermediate, and they're saying, okay, how can I apply this to my training? And the short answer is you can't. But it's a nice scientific question of let's figure this out, and it's going to take a while.
That's well said, Brandon. I think you also said a few things that I want to really cue in on. I think where I'd like to start is that there is risk because these factors are so interdependent, that we could easily, if we don't do what Dr. Schoenfeld saying of not being too strong on our conclusions, make the same mistake that was made in the 90s and in the 2000s with the hormone hypothesis that, okay, every time you lift weights, because we're not actually a mechanical machine, we're a biological entity, if we're producing progressive overload, we're probably inducing some level of muscle damage. It's unavoidable. We've got these contractions occurring. And at the same time, the contractions require fuel. So that means there is metabolism happening, if you will, when you're contracting. So there is an obligatory level of metabolic fatigue to some degree, even if it's very small, and an obligatory level of damage anytime you're producing progressive tension overload. So these are intrinsically tied factors. So they're always going to be there. So inducing muscle growth, there's this correlation causation confusion where how do we actually know what is the causative factor there? I think we're probably all pretty comfortable saying that there is no damage or metabolic stress without lifting weights, at least for the kind of levels we're talking about, or at least with contraction, we can say, in a real world situation. But it is really interesting when you start looking at some of these potentially animal models or non in vivo models where injecting lactate to see if that produces muscle growth independently, or using a drug that causes muscle damage. I think that's really where there's some interesting lines of inquiry, because there's a difference between a permissive pathway or an additive pathway or indirect. For example, we can explain a lot of these applied things away if we wanted to play devil's advocate, like blood flow restriction training. We can say, well, yeah, that's not actually metabolic stress causing new or different hypertrophy. It's just producing early fatigue so that you have to recruit high threshold motor units earlier. It's the same thing as lifting heavy and just kind of circumventing all of the quote unquote early reps in a set. Or we could say, well, it's not the muscle damage, it's the overload, So I think it's important for us not to make the same mistakes that were made with the hormone hypothesis. And I guess the next question I would have for you guys is, what is the next frontier? How do we prevent those mistakes, and what data do we need, and what kind of designs might be required to answer those questions? Juha, let's start with you, buddy.
Yeah, I think one of the most important things was that you explained about this untrained and trained. So when we have untrained people starting to train, like this, when they start to train, there is this homeostatic response, so high protein synthesis response, perhaps some protein breakdown and homeostasis response to correct those damages that we have in our muscle fibers because of this novel stimuli. It's totally different than when we have trained for like 15 years or 45 years, like Brad has trained, something like that. Is it 45? Yeah.
I started when I was 1, so yeah.
Yeah. So it's a different situation. So how our muscles can then still grow when we have this background of years of training. So do we have can we draw really those same conclusions from those untrained studies when compared to those trained ones? So I think it's and also this mice are not men type of a thing. So we have to be careful. So we have to just understand these things, and I think it's important that these people who are doing these mechanistic studies with animals and some applied biological studies in humans, that they also train themselves so that they know that what is this whole thing, that you can get hypertrophy by training some with different types of ways, and then when you get bigger, it's more difficult to grow even more, and so on. So I think it's really important that we are combining this applied side to the mechanistic side. So I think that's perhaps the most important thing that I would suggest, and I think this is what we are doing now here in this podcast and some other people as well. So with Brad, we have tried to combine this applied side to really hi-fi mechanistic side. So I think that's quite important. So you have to lift to understand these kind of things.
Yeah, that speaks to what I often call the quote-unquote chain of research. Something I talk to my students about a lot is that you can make errors if you go directly to application from purely mechanistic data without doing that applied research step. So for example, if we uncover all these mechanisms, but then we don't put them into play in a training program, I personally think it's too early to say, hey, you should do some training that is damage dominant, you should do some training that's metabolic fatigue dominant, and you should do some training that's tension dominant. I think that makes sense theoretically. We really need to test it, because we have seen, although some of the papers you mentioned, Brad, like DeMoss, can't tell us that damage isn't important. What they have told us is that if you do a lot of damage, that becomes the priority, and your body has to focus on regeneration first. So because like we said, everything comes from the initial tension stimulus. So if we end up subverting mechanical tension with too much metabolic stress or damage, or too much focus on that, now all of a sudden we're kind of getting the cart before the horse, if you will. So in my opinion, it's a case of, right, we know that all these factors play mechanistically, but we can't yet say, what does that look like in our training? Is just standard kind of strength training that produces progressive overload, is that inducing enough damage? Does that induce enough metabolic stress? Or would it give us additional hypertrophy if we train in other ways? And I think that's what's, Juha, you pointed that out really well. We've got to start studying whether or not programs based on the theoretical model produce enhanced outcomes in trained lifters. Because if they don't, then it's all a really cool understanding, but it hasn't changed our practice.
It's all cool story, bro. I mean, my takeaway, Eric, is what Brad said about the 300% muscular growth in the quails, where their wings were attached like that, applying it right at the gym. I'm just going to hold this for a month. 30 days, 300% growth, done.
Oh, so I wasn't listening. I thought you had to hold 300 quails to grow your lats or something. I don't know. I already ordered a bunch of quails online. I don't
Quail
know if I
eggs.
did that right.
There actually is someone...
Yes.
I just want to say, there is actually an individual who I will not name, who has used his physique to sell a lot of different things, including the frog pump, which you guys might be aware of, that was marketing for a while duck eggs for their extreme hypertrophic response. Anabolic potential. Yeah, so we're not that far off. The five of us combined launched quail eggs. We got a business.
If you get one take home from this podcast, it's eat quail eggs.
Yeah.
Yeah, and also if you want to increase the size of type 2 fibers, which are white, you should eat white meat as well.
I'm going to take this, Eric, I'm going to take it from somewhat of a different angle. That personally, I don't think we're at a point where we can necessarily take mechanistic factors and try to apply them to training. And I don't do that certainly in my own practice. I think you go the other way, and that you look at the applied research that we have on the different variables on manipulating, so volume, load, frequency, et cetera. And ultimately, that's going to fit into the mechanisms anyway. I do think that, so let's say we're talking load, we know that on a whole muscle level, it really does not matter whether you're training with heavy loads, moderate loads, or high loads. But there might be benefits from which I mentioned before, a fiber type specific level, targeting type 1 or type 2. Again, more research needs to be done. But in the absence of evidence, and certainly with a good theoretical rationale, and assuming your goal is to maximize total muscle growth, you train across a spectrum of rep ranges. Well, that's going to get you metabolic stress, and it's going to get you mechanical tension, and it's going to get you muscle damage. So ultimately, you end up hitting these things anyway. With higher volumes, we know generally comes more metabolic stress, depending on the repetition range you're working in. So we know that volume is a driver of hypertrophy, and that however you're going to structure your routine volume-wise, that ultimately, if you're going to be doing somewhat higher volumes, you're going to be inducing metabolic stress, and perhaps even more muscle damage. So I think at this point, I think as Juha had mentioned, maybe Brandon as well, that ultimately understanding the mechanisms will allow us to better, when we do have that understanding, to better target our routines. We're not there yet. And without being there yet, I think you don't try to force that, and you then take the other way.
Well said. Yeah, exactly. I think the chain goes both ways. We can learn from practice some hypotheses about what we should check in research, and that's both at the mechanistic level, then feeding back into applied, which then feeds into what we actually do in the gym. So I think that's a really good perspective. Thank you, Brad.
I mean, definitely echo their thoughts, and I'll throw a new kind of curveball at us that we haven't really talked about. And that's one of the things that I really love is, you go into the gym and someone's doing a protocol or a program and they're getting great gains, and you try it and you don't get as good gains, and you're like, wait, what? I thought we had the maximal program. What happened? So there's also this underlying response variability, is what we call it, right? So I think the future, once we figure out these mechanistic things, and while we're figuring out the applied part, is saying, okay, can we? And we're nowhere near close to this. Can we somehow tell, however we can do that, who's going to respond best to a program, or maybe like this is the point when you need to switch, or maybe it has to do with enjoyment or consistency and other stuff too. There's lots of biobehavioral stuff, but just applying more of a big picture of like this is training, yes, but it's training for different people, but we have to look at group means to tell us answers, right? So there's just a lot to unpack there. So I'm hoping, and it'll happen for sure over the next decade or so. We'll figure out, what these mechanistic things are, how to apply them, and then how to apply them to certain people.
Love it. So I think one thing that I've seen, and Brad alluded to this, is I think because of the nature of damage and metabolic stress being intrinsically tied to mechanical tension, and it's so easy to mistake correlation for causation there. And we've seen the prior history and danger of us going down that path for a couple decades with the hormone hypothesis. I think that's why we see these blog posts, or sometimes they're researchers and not just kind of your standard layman who is interested in science will make these statements of metabolic stress or damage is not related to hypertrophy, which I would agree is probably a little too strong to say. But how would we? What is the way we could definitively know, if we could wave a magic wand and have magic research to really get an idea? And I know this is actually a challenging question, because if we knew the easy answer, we'd already done it, right? How would we be able to disentangle muscle damage or metabolic stress from mechanical tension to get an idea of what was if it was true that they weren't related, or were so minorly related it was just kind of secondary?
If I knew that, I'd be carrying out the study.
I love it.
Yeah. So you'd have to block two of them and try to figure out the third one. But since they're all connected, that would be hard because there's some inherently confounder there. But if we could figure out a good start, right, would be maybe targeting some of the metabolic stress metabolites and saying, okay, we're going to block these in humans safely, and just seeing what that does, right? Or saying, we're going to use a protocol for muscle damage that's ramping, that's super duper 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 slow, and these people don't actually get muscle damage for some reason.
Yeah, it's really complex. So, for instance, this metabolic stress phenomenon, so we can detect thousands of molecules that are affected by exercise. So then we would need to individually manipulate those molecules and say, okay, this one, this one, this one. So of course there are some metabolic stress metabolic stress related metabolites that will be anabolic, and some of those will be catabolic. So we already know that some of the energy stress related metabolic stress metabolites and pathways, they are catabolic. So if we induce too much of this energy stress related thing, for instance, we will do too high volume of exercise, like we would do like continuous exercise of 24 hours, then we will induce atrophy. So even though we would have really high metabolic stress, so we know that marathon running, for instance, will induce atrophy. If you just continue to do that, you will decrease your muscle fiber size, even though there is some damage and even metabolic stress sometimes. So if we have too high of a stimuli, then that may be problematic. Of course, like Brandon and others, you have written that there are inflammation and other complex things that make it difficult. But coming back to this issue, we have been discussing about this with Brad. Can we do that in humans? And so far it's really difficult. But we have bought this cell system so that we can stimulate muscle cells, like individual muscle cells, to do sort of exercise, and also we can stretch them. So I think with these kind of systems, it's possible. So for instance, we can induce just pump per se to muscle fibers. Then we can see whether this can induce something, for instance. So those things we can check some of those things. But then applying those to humans, then that's going to be difficult. So, but anyway, I think if we do a lot of those, enough of those studies, like with cells, with animals, with humans, and so on, in the next 10 years, maybe we will write another review and we can figure out whether we have some new answers, maybe with this group or some other group, and let's see. Yeah, but that's really difficult.
As remember, we pointed this out in our paper, the paper I did with Juha and Henning. There are many metabolites. Generally we're thinking of lactic acid, and inorganic phosphate probably is the other one, but calcium would be another very potent one to look at as a metabolite. There's the calcineurin pathway, the calmodulin pathway. Both of them have been shown to regulate muscle growth. They are anabolic stimulators. Now, there's other research that shows in certain levels they actually stimulate more aerobic metabolism, so it's still not clear. Even that, just in terms of the pathways that are stimulated. But that's why, again, trying to tease out just one metabolite, maybe we'd try to look first at lactic acid would be one, do some type of buffering. Again, maybe Brandon or Juha can figure something out, and I'd love to collaborate on that. But even if we do that, it would not necessarily that we would just then say that aspect of metabolic stress, but there's certainly many others that would need to be considered. Phosphatidic acid, another metabolite that has clearly been shown to regulate, it has downstream effects on P70S6K, which is again, a primary enzyme for protein translation, muscle protein translation. So anyway, these are all nuances that often are not considered, that need to be considered, that are very important. It's a very nuanced topic that is way too often oversimplified. And I know that's the way in the current internet environment and social media. That's what happens when it gets into the lay public. They're going to look to simplify things and to kind of package it into a T Nation article. But it's much more complex.
And it's also difficult because even with all these complexities, where we get when you guys write that second paper another decade from now, which I will vociferously read just like I did the first one, is that it may end up being something quite similar to what we're already doing. Train across the spectrum of rep ranges, progressive overload, do an appropriate volume, and like Brandon was saying, figure out where your individual thresholds lie. And, I think that will probably be the primary take home. But this is Iron Culture. We're talking about people who are willing to put in the extra work to get a level of gains beyond what they might have had that's not even noticeable to most people, but that was worth it, damn it. So I think
still real to me, damn
that's it. Absolutely, because we're muscle nerds. No. So I think I think that's a really important perspective, is to understand that that absence of evidence is not evidence of absence, or I said that backwards, meaning that just because we don't have the data to say something is connected doesn't mean we know it is not. And that's a common mistake that I think is just part of science, because we're always technically disproving things, that just because we don't have a methodology that allows us to disentangle this stuff doesn't mean that it isn't actually happening that way. And just because the output and the application we kind of have a feel for, progressive overload, do enough, train across the spectrum of rep ranges, doesn't mean that there isn't more going on. So yeah, this may not change what you're going to do on Monday when you step in the gym, which is bench, but it certainly is something to think about for us in the future, so we can move towards better gains. So this has been awesome.
Yeah, one
ahead.
last thing, Eric. I know you mentioned that like some researchers had taken the hard positions, and that really, to me, is the more important thing to tackle at this point, because I think there is. I've seen. I think it's unfortunate that a lot of times researchers become opinionated and then cherry pick. They tend to be prone towards confirmation bias in presenting their. Basically, they act like a lawyer and they present their case for something. When what we need to do in these instances, and I think I would hope people feel, and what we tried to do in the paper that we wrote, the sensors and stimuli paper with Henning and Juha, that we tried to take both sides. That we're not in this. We don't have a position. I'm happy whatever happens. For me, I don't care if ultimately the metabolic stress or muscle damage does or doesn't turn out. I just want to know. Really, true scientists seek the truth. And when you have both sides of the coin, we don't want to position things to try to make an opinion for ourselves. We want to recognize the other sides of the coin here, and thus then say, all right, here's the weight of the evidence. And then we could have differences in opinion. But what I've seen all too often is people taking this side and not presenting this side. And that then skews it. The general public doesn't know. And they're the ones who are going to be impressionable when a very popular either researcher or fitness professional is taking this type of position. And I just hopefully, one person get up on my soapbox, but one of my hobby horses is to try to get this into the population or to the consumers, that when you're always be skeptical. So just because I say something, or maybe if Eric says something, you can believe that. But the rest of us, it's
I'll echo
that's
that.
Eric can agree with that. Yeah.
But in all seriousness, if just because a expert says something, we have certain knowledge and credence, but you still want to be skeptical. And there are people that become hardened. They can be very good researchers, but they can be hardened in their opinions and not be presenting the full picture. So don't just follow the crowd. Try to become an educated consumer. That's my soapbox.
And avoiding
well
black and white thinking is the thing that you.
Most importantly,
that's really well said.
I was going to say, I like how all your answers came back to the idea that if only we can experiment on live humans, we'd probably get to the bottom of things. And I just want you to know that after this podcast comes out, I'm sure we're going to have some willing subjects, some willing participants that will sacrifice themselves for the greater gains of good. So we might have the answers after all.
Yeah, finally,
yeah.
And also, I'll just put it out there, like, if you want to go to the premier sports science university in Finland, you can head out to Jyväskylä and do some study. And also, Brad Schoenfeld is now taking graduate students, folks. This is if you're listening and you're like, well, I got an idea, I want to do this. There's some people to contact where you can get involved and actually be a part of the solution, rather than slagging us off with your Instagram handle because you think damage doesn't matter or you think it does. So get out of the Instagram comments, get into your graduate research is what I'm saying. This is a plug for hypertrophy researchers of the future.
Look, we learned everything we needed to know from Arnold's Encyclopedia of Bodybuilding 40 years ago. Everything else is just, frills. I will say, though, that I think it's cool, and I spoke previously about this, how this is a field where you could be so closely connected with those at the top, with experts, with those trying to make a difference. If you wanted to send, for instance, someone like a Brad or a Juha or Brandon a message, you can, and you'll get a response, versus other disciplines or other fields where it might just be so hard to reach that point or even get on that path. So I think it's cool how accessible in ways fitness can be, or how it's become.
Absolutely.
No.
So I'll just open it up to the three of you. Any closing thoughts, things that we didn't present well enough, or that you think people should keep in mind on the topic of hypertrophy before we hear where people can find out more about you each?
I got nothing.
Nothing for Brandon. He's hungry. Juha, you got anything?
Right. I'm hungry.
I'm sleepy. It's almost midnight here, so.
Love it. Brad, any other closing remarks?
So the one thing that I would close in, and I would say that you can make very good gains on a very simple, basic routine and very low volume routine. Really, when we're talking about the science of muscle growth, it's about optimizing genetic potential. So when people hear this, if you want to just do a basic HIIT type routine, 1 set to failure, 3 days a week, kind of the old Arthur Jones or Ellington Darden routines, you can achieve a large percentage of your potential. But if you will never, in my humble opinion, based on not only my own anecdotal evidence, but the totality of research, to optimize, to get that extra 10, 20%, whatever it is, that's where you need to really start to apply scientific. And the final thing is there's a very large inter-individual variability. I'm not sure if I kind of caught the tail end if you were getting into that, Brandon, when I had to get my charger here. But it's really important to know that just because something works for one person, it's certainly not going to work for another, and that the more of a poor responder you are, because we see very large variations. When I do a study, I'll have some people are gaining 0 or even losing a little muscle, and some people are gaining 20, 30%. Why does one gain more than another doing the exact same thing? Now, we're not looking at we do food diaries, but the food diaries, first of all, are somewhat inaccurate. And we don't try to correlate those. It's very difficult to try to say, right, you ate this and you ate this. And so I think a lot of it, you just have to chalk up to certain genetic variation, lifestyle factors. And that means that, look, as a personal trainer, there was never anyone that I was not able to get results with. So the people that in my studies are not growing, it's because that didn't fit their own personal genetic code, if you will, for bringing out their best. And that's why ultimately everyone is an N equals 1. So you look at research, you look at the studies, use that as a guideline, and then you have to really experiment from there and find out what works best.
Well said. Yeah, the importance of interindividuality cannot be overstated enough. So I think that's great. So thank you for those closing remarks. And Juha, where can people find you if they want to learn more about your research or what you do or your book?
Yeah, people can find me from gym, obviously, or metal gigs or something like that. But yeah, but anyway, I have a blog. It's in Finnish. It's called Muscle Doctor Blog, but it's in Finnish, so it's Lihastohtori. It's in Finnish, so you cannot really find me there. So you can just Google me and find some of my work in research. I think that's most important, and my website, university website, perhaps. And if there are Finnish listeners, then you can find my website and books and those kind of things. Yeah,
fantastic.
Probably is honestly 0.5% of the audience. So you think like no, it's like no. There's probably this video. There's probably 200 people from Finland, watching it. So
I think after this there are much more. So I promise.
Yes. We're maximizing our Finnish audience.
Yeah.
Happy to hear it. Love it.
Yeah.
Brandon, how about you, my friend?
Facebook's probably the most active for social media. Strength Guy's website, scifit.net. It's kind of the science of communication and pretty much everything right there. And then if you can always email me, Brandon at strengthguys.com if you want to chat or something. I try to help out students all the time. Or if you just want to talk science, I'm down.
Beautiful. And last but not least, Brad.
All over social media, Twitter, Instagram are the primary ones that I'm using now, but Facebook as well, although their algorithm sucks. So I've devalued that in terms of priority. My ResearchGate page, I have close to 200 publications now, if not more. And most of the research is freely available on my ResearchGate page. So you can check that out. I have a book, Science and Development of Muscle Hypertrophy, that delves into a lot of the stuff that we're doing. And certainly shoot me a direct message if you're interested in pursuing a graduate degree in New York, the best city in the world.
It is a phenomenal city.
Right. Awesome. Thank you guys so much for your time. We really appreciate you being here. Omar, I'm going to let you handle these closeouts because you're the best at it.
If you've made it all the way to the end of this podcast, you either have no life or you're very interested in the science of hypertrophy, which is a noble pursuit. We have weeded out all the shut up and lifters, and hopefully you've taken away a lot of information. Everyone's information will be linked in the description. Thank you for listening. You can help us out, the Iron Culture Podcast, by leaving a rating review if you're on iTunes. If you're on YouTube, feel free to give us either a thumbs up or a thumbs down if you didn't enjoy it. But let's face it, it's only going to be thumbs up. And leave a comment in the comment section, and we'll do our best to respond. And we'll see everyone in that next episode of Iron Culture on Monday.